Good afternoon, or good morning, or good evening, depending on wherever you may be. We are so glad you could join us. Welcome all to our Clinical Pharmacology Considerations for Novel Therapeutic Modalities webinar. My name is Corey Adair, and I am with the Small Business and Industry Assistance, or SBIA program. I will be your host for today's session. Our SBIA program operates within the Division of Drug Information, or DDI, which in turn resides within FDA's Center for Drug Evaluation and Research, or CDER's Office of Communications, or OCOM. Here are the learning objectives for today's session. Now, before we begin, we encourage you to connect with us. For your convenience, we've included QR codes to access SBIA on LinkedIn, X, YouTube, and via email updates. Scan the QR code with the camera on your cell phone. This will provide you with a link to SBIA on the platform for the QR code that you scanned. It's great to stay connected with SBIA social media platforms to learn about upcoming webinars, conferences, and regulatory information, and also to find recordings of past webinars and conferences. Now, let's turn our attention to some logistics. Please note that this event is 100% remote, in addition to being 100% virtual. Our speakers and panelists, not to mention our SBIA team and technical staff, are in separate locations. As such, there may be some internet issues, but please bear with us. And if in fact we encounter those, we will try to get them resolved as quickly as possible. We invite you to submit your questions to us so that we have a vibrant and productive Q&A segment, which is so crucial to these events. This is a learning opportunity, and I encourage you to take full advantage of the expertise we offer here. When you do submit your question, please identify that individual presenter by prefacing your question with the speaker's name as shown here, @Raj. Please keep in mind that while we may not get to your individual question, they are always very helpful as we plan for future events. Again, SBIA will send a thank you email to attendees within 24 hours after today's session. Recordings of today's webinar will be posted to the SBIA webpage around 10 business days post-webinar. Now, I'll take a moment to call attention to our upcoming events. On December 10th through 12th, SBIA will host the FDA Clinical Investigator Training Course, or SITC 2024 conference. On January 22nd, SBIA will host the BSUFA 3 Regulatory Science Pilot Program Updates webinar. On January 28th through 29th, SBIA will host the Knowledge Management and Modernization of Regulatory Quality Assessment and Submissions at FDA conference. And on April 9th through 10th, SBIA will host the Generic Drugs Forum, or GDF 2025 conference. We also invite you to visit our CDER Learn webpage, where you can link to SBIA tutorials, along with other educational resources, many of which offer CEs. That can be found at www.fda.gov/CDERLearn. Now, it is now my pleasure to introduce our first presenters of the webinar. First, we will hear from Dr. Rajanath Madabushi, Associate Director in the Guidance and Policy Team within the Office of Clinical Pharmacology and the Office of Translational Sciences within CDER at FDA. Then we will hear from Dr. Hobart Rogers, Captain of the United States Public Health Service and a reviewer in the Division of Translational and Precision Medicine, also within the Office of Clinical Pharmacology and the Office of Translational Sciences at CDER at FDA. Then we will hear from Dr. Anuradha Ramamurthy, Master Scientist and Policy Lead in the Guidance and Policy Team, also within the Office of Clinical Pharmacology and the Office of Translational Sciences at CDER at FDA. Now, please join me in welcoming Dr. Raj Madabushi.
Thank you, Corey. Greetings, everyone. My name is Raj Madabushi. I'm an Associate Director for Guidance and Scientific Policy in the Office of Clinical Pharmacology at CDER. Before we do a deep dive on the specific topics, I will provide a brief overview on the changing landscape of therapeutic modalities and its implications for today's SBIA webinar. Historically, in CDER, small molecules have been the mainstay modality of approvals. And as such, to aid in the drug development, we have developed many regulatory guidances. However, over the past decade or so, we are seeing a shift in the landscape with novel therapeutic modalities getting approved. These include biologics, oligonucleotides, antibody-drug conjugates, bispecifics, and many more. To emphasize this changing landscape, I'm showing you here the CEDAR approvals in 2019. You can see 3/4 of the approvals were small molecules, with some newer modalities beginning to get approved. Now contrast that 3 years later in 2022, where the number of small molecule approves dropped to just over half of the CEDAR approvals. And now we are seeing more newer modalities being approved. It should be noted that the newer therapeutic modalities have distinct attributes and thus may need different considerations for development. Here, I am providing you with a comparison of various attributes for small molecules, oligonucleotides, and monoclonal antibodies. While this is not an exhaustive listing, one can clearly appreciate the various differences that are likely to impact the clinical pharmacology characterizations. Clearly, those that can affect the pharmacokinetics and pharmacodynamic aspects. These differences highlight the need for modality-specific considerations to inform drug development. Given the emergence of newer modalities, we have been gearing up to develop targeted regulatory guidances to assist with their development. Here is a timeline of some of our efforts. Our initial efforts were focused at addressing the immunogenicity concerns for monoclonal antibodies. And over a period of time, we have focused on developing clinical pharmacology considerations for specific modalities such as bispecifics, oligonucleotides, monoclonal antibodies, antibody-drug conjugates as well. Today, our focus is on two final guidances published earlier this year. The clinical pharmacology considerations for antibody-drug conjugates and the clinical pharmacology considerations for the development of oligonucleotide therapeutics. With that, I will pass it over to Dr. Bart Rogers to get us started with oligonucleotides.
Well, thank you, Dr. Matabushi. My name is Bart Rogers, and I'll be joined by my colleague, Dr. Anu Ramamurthy, and today we're going to be discussing our recent final guidance, the clinical pharmacology considerations for the development of oligonucleotide therapeutics. And I'd like to thank everyone for joining us today. Now, oligonucleotide therapeutics are considered an emerging therapeutic modality and are of keen interest to the FDA. Importantly, I want to recognize that they represent a very diverse category of therapeutics, from mechanism of action, various chemical modifications, structure, delivery strategy, conjugation, etc. And it's important that we point this out because all of these can certainly impact the clinical pharmacology considerations and assessment of these products. Importantly also is that they are somewhat unique compared to small molecules and biologics. They tend to have a very long pharmacodynamic half-life, limited PK exposure, often a disconnect between PK and PD that we're used to thinking of with small molecules. So some unique considerations for these products. So what exactly are RNA oligonucleotides? These are modified nucleic acids that are typically designed to bind to a complementary RNA sequence utilizing a Watson-Crick-Franklin base pairing. Now they've been synthetically modified to increase specificity of binding, to slow degradation from endogenous endonucleases and exonucleases, and avoid immune recognition or immune stimulation from our immune pattern sensing receptors. So there are various means of this synthetic modification. For instance, the backbone can be chemically modified. Typically, we will see either a phosphorothioate or morpholino, 6-member morpholino backbone. as well as synthetic modifications at the 2' position of the ribose sugar. Fluorination, monoethoxyethyl are common substitutions here. If you'll denote on the bottom of the screen, this is a depiction of the two most common moieties in this field. One, the single-stranded antisense oligonucleotide on the left, as well as the double-stranded siRNA on the right. So why oligonucleotide therapeutics? When thinking about the central dogma of molecular biology, DNA, RNA, protein, for the most part, small molecules of biologics have only been able to mitigate disease at the level of protein. Whereas now with oligonucleotide therapeutics, we can work a level upstream to mitigate disease at the level of the RNA. Importantly, oligonucleotide therapeutics are able to target the cause of many genetic disorders that were previously considered undruggable. They utilize endogenous cellular machinery. They can increase expression of needed proteins for an agonist-like effect or silence expression of pathogenic proteins. In addition, they're able to be quickly synthesized using a similar chemistry in some of the chemical synthetic modifications that I previously mentioned. To date, the FDA has approved 17 oligonucleotide therapeutics from fomivirsen in 1998, currently to imetelstat this year. Of these, there's been 11 antisense oligonucleotide products and 6 siRNAs. You also note here in blue, a number of these products have been approved on surrogate endpoints and are waiting confirmation in the post-marketing space. When looking at the IND submissions per year, I look at this based on really 3 time points. The period of 1992 to around 2002, the oligonucleotide therapeutic field was met with a lot of failures, very, very marginal investment in this field, and very high risk. And then we started to see some success in the IND space from approximately 2003 to 2013, advancements in chemistry in addition. And we did see some breakthroughs here. We saw the first drug mepomersen approved since 1998. And then really what I'm calling the modern era of oligonucleotide therapeutics from 2013 really to the present day. We've just seen an explosion in growth in the field in terms of IND submissions. The chemistries have been de-risked to an extent, and larger pharma has also begun to invest heavily in this new therapeutic moiety. So recognizing this important impact of oligonucleotide therapeutics, we try to prospectively anticipate some of the common challenges and based on our data, internal data, put together a guidance on the clinical pharmacology considerations regarding oligonucleotide therapeutics. And our finalized guidance was published in June of this year. Now, I want to point out that this guidance, while it does hone in on 4 topics, it does not encompass the full arena of all clinical pharmacology topics surrounding these oligonucleotide therapeutics. However, we felt these were the low-hanging fruits where sponsors commonly were seeking advice, and we wanted to prospectively provide evidence for that. Here, I'll be discussing 2 topics, the QTc prolongation and immunogenicity risk, and then my colleague Anu Ramamurthy will be discussing organ impairment and assessment of DDIs, drug-drug interactions. So when looking at QTc prolongation and the evaluation of pro-arrhythmic potential, it's very important that sponsors continue the assessment of QT prolongation risk, and this should be conducted. And there's a number of ICH guidances we want to point sponsors to allow them better assessment and how to go about the assessment of the QT prolongation and pro-arrhythmic risk of these oligonucleotide therapeutics. Now, it's also important that all proposals and assessments should be adequately justified and discussed with the agency in a prospective fashion. So again, we encourage sponsors to meet early and often with the FDA when discussing these matters. And importantly, the timing and extent of QT assessment will often depend on the overall risk-benefit profile of the oligonucleotide. For instance, is this a well-established, well-characterized chemistry, or is this a novel chemistry where we don't have a very good understanding of the QT risk and pro-arrhythmic potential? Now I want to talk about the second topic is assessing immunogenicity risk for oligonucleotide therapeutics. We ask that sponsors follow a risk-based approach in the immunogenicity assessment of oligonucleotide therapeutics, and they can look to the immunogenicity assessment for therapeutic protein products published in August 2014 for further details. In addition, we really want to highlight the importance of establishing immunogenicity assay early in development and validating the samples early in development, and in addition discussing with the agency early in development. Now, immunogenicity sample collection should coincide with the PK and PD sampling time points for the oligonucleotide therapeutic. And as I previously mentioned, given the typically long pharmacodynamic half-life, the sampling time points may be spread across months. given that many of these oligonucleotides are dosed on a monthly basis, not a weekly or daily basis. Now, based on the risk assessment, it may just be adequate to bank samples in development for later testing if the oligonucleotide is considered to be low risk. Again, this should first be discussed with the agency, and sponsors should discuss immunogenicity risk assessment and how it will inform their clinical immunogenicity assessment moving forward with their clinical development program. With that, I'll turn it over to my colleague, Dr. Anu Ramamurthy. Thank you.
Thank you, Dr. Rogers. Hello, everybody. My name is Anu Ramamurthy. I am a master scientist in the Office of Clinical Pharmacology, CDER, FDA, and I will be covering the next topics in the clinical pharmacology considerations for the development of oligonucleotide therapeutic guidance. The first topic is characterizing the impact of hepatic and renal impairment on pharmacokinetics, pharmacodynamics, and safety. To determine the appropriate approach for characterizing the impact of hepatic and renal function on the PK, PD, and safety of the oligonucleotide therapeutics, the sponsor should identify the role of the liver and kidneys in the disposition, elimination, and drug response of the oligonucleotide therapeutic based on preclinical and early phase 1 clinical data. This early PK/PD characterization of the oligonucleotide therapeutic, along with safety and tolerability information, should be used to inform the enrollment of participants with a full range of hepatic and/or renal function in the late phase trials. In addition, in participants with organ impairment, it is also important to consider the impact of changes in the expression and turnover of one, the target of the drug, and two, in the case of a conjugated oligonucleotide therapeutic, the target of the conjugate. That is, for example, receptor expressed in the liver that allows for the targeting of the oligonucleotide therapeutic to the liver. When the oligonucleotide therapeutic is not predominantly renally cleared, or the oligonucleotide therapeutic does not target the liver, the late phase clinical trials should generally include patients across the full spectrum of hepatic or renal function. This approach allows us to accumulate information on safety and efficacy in patients with hepatic and renal impairment in the patient population that will eventually receive the drug if the drug is approved. In this case, dosing should be based on information from non-clinical studies and early clinical experience. The sponsor should provide justification if participants with impaired hepatic or renal function are excluded from late phase clinical trials. Now let's look specifically at renal impairment. When the oligonucleotide therapeutic is not predominantly renally clear, the sponsor should enroll subjects with a full range of renal function in the late phase trials based on information from non-clinical studies and early clinical experience. When the oligonucleotide therapeutic is substantially renally clear, that is greater than 30% is excreted unchanged in urine, further characterization of the impact of renal impairment is generally recommended following FDA's renal impairment guidance. There are several different strategies that can be used to study the impact of renal impairment on drug exposure and response. One option is the reduced study design. This can be considered to assess the impact of severe renal impairment or kidney failure not on dialysis on the pharmacokinetics, pharmacodynamics, tolerability, and safety of the oligonucleotide therapeutics. When applicable, the study should have long enough follow-up period to enable adequate characterization of the pharmacodynamic effect. With appropriate justification, other alternative approaches can also be considered. The findings from such study can inform any additional characterizations as well as the inclusion and exclusion criteria for subsequent late phase trials. Next, moving on to hepatic impairment. It is important to understand the role of liver in the disposition and elimination of oligonucleotide therapeutic. When the oligonucleotide therapeutic is not liver targeting, the sponsor should enroll subjects with full range of hepatic function in the late phase trials based on information from non-clinical studies and early clinical experience. When the oligonucleotide therapeutic targets the liver, that is, the pharmacological target is in the liver or there is active targeting to the liver, the sponsor should consider characterizing the impact of hepatic impairment. In this case, sponsors can consider alternative approaches that allow for sequential or adaptive enrollment, incorporating results from early phase studies on tolerability, safety, and pharmacodynamics. This information can be used to facilitate enrollment of subjects with a range of hepatic function in the late phase trials. Some additional considerations. Because changes in organ function can result in pharmacodynamic changes that are independent of pharmacokinetic changes or not reflective of pharmacokinetic changes, whenever appropriate and feasible, the sponsor should conduct pharmacodynamic assessment. When appropriate, population PK/PD modeling can help assess the correlation between organ impairment and pharmacodynamics, along with other data such as biomarker, safety, or efficacy data. In order to do this, it is important to enroll sufficient number of participants over a range of organ function across the entire drug development program to obtain meaningful data in all categories of organ function, unless there is adequate justification such as safety concern. Moving on to the next topic, considerations for assessing drug-drug interactions. I will touch on three topics: oligonucleotide therapeutics as substrate, as modulators. These are pharmacokinetics-based drug-drug interaction. And last, I would touch on pharmacodynamics-based drug interaction. First, oligonucleotide therapeutics as substrates for cytochrome P450 and transporters. Oligonucleotide therapeutics are not typically metabolized by cytochrome P450 enzymes. These are primarily metabolized by exo and endonucleases or are chemically modified to resist degradation. Therefore, the disposition of oligonucleotide therapeutics is not anticipated to be affected by inhibitors or inducers of cytochrome P450 enzymes. Additionally, modulation of efflux transporters, hepatic uptake transporters, or renal uptake transporters, or renal efflux transporters are generally not anticipated to have a significant impact on the pharmacokinetics of the oligonucleotide therapeutic. Now, moving on to oligonucleotide therapeutics as modulators of cytochrome P450 enzymes and transporters. Based on current experience, oligonucleotide therapeutics either do not modulate or very minimally modulate the major cytochrome P450 enzymes and transporters. However, an overall recommendation for specific type of oligonucleotide therapeutic, example, based on chemistry or based on delivery strategies, cannot be provided at this time. So evaluating the drug-drug interaction liability of oligonucleotide therapeutic as inhibitor and inducer of cytochrome P450 enzymes or transporters usually begins with in vitro assessment. The sponsor should provide adequate justification if in vitro assessments of the potential for oligonucleotide therapeutic to affect cytochrome P450 enzymes or transporters are not conducted. And if the in vitro studies indicate that the oligonucleotide therapeutics could modulate the P450 enzymes or transporters, the sponsor should consider clinical studies to evaluate in vivo drug interaction. There may be other possible mechanisms for interaction between oligonucleotide therapeutic and the cytochrome P450 enzymes or transporters, or the modulators of P450 enzymes or transporters, for example, by interfering with the synthesis or degradation of heme or cytokine. These mechanisms should be considered based on the pharmacology of the oligonucleotide therapeutic. The last topic is pharmacodynamic interaction. Oligonucleotide therapeutics can exhibit pharmacodynamic interactions with concomitant drug when the pharmacological effect of one drug can be altered by that of the other drug. Example, drugs with shared mechanism of action pathways. Because such interactions are very unique to the oligonucleotide therapeutic in question, we encourage the sponsors to consult with relevant review division regarding assessment of pharmacodynamic interactions. Thank you for your attention. This was a high-level overview of the final guidance for the industry titled Clinical Pharmacology Considerations for the Development of Oligonucleotide Therapeutics. Now I will pass the mic back to the moderator for the Q&A session.
Thank you again to our presenters for the excellent presentations. We will now begin our first question and answer panel. If you haven't had a chance to enter your question into the Q&A chat box, please do so now. We will answer as many questions as time allows. So, to go to our first questions today, our first question is directed towards Dr. Rogers. And with this, I would also like to introduce two additional panelists that will be on this Q&A chat today as well. First, we have Dr. Oluseyi Adeniyi, who is a reviewer within the DTPM within OCPT in the Office of Translational Sciences at CEDAR, as well as Dr. Wendy Wu, who is a senior pharmacokineticist within the Division of Applied Regulatory Scientists in the OCP, as well as in the Office of Translational Sciences at CEDAR. Now we will begin with our first question here to Dr. Rogers. This question is, what clinical ADME, DDI, or mass balance studies are requested for oligonucleotide drugs?
Yes, thank you for the question. In general, oligonucleotides, again, represent a diverse category of therapeutics. You want to point that out. If an oligonucleotide is considered well characterized, meaning there's an approved therapeutic modality with a similar chemistry and there is right of reference, typically ADME studies may not be necessary. Now, regarding mass balance, we do tend to notice how oligonucleotides distribute, so mass balance are not typically required. What can change all this is if there is a novel chemical moiety, a new chemical moiety added, a new 2' substitution, for instance. This can change the ADME characteristics as well as the need for a mass balance study.
Excellent. Thank you for that response. Another question for you, Dr. Rogers. Does this new guidance on oligonucleotides apply to DNA phosphorylsiolate ODNs or ODNs TLR9 agonists, which do not have a mechanism of action that is based on pairing against a target sequence, or only to an RNA ONT?
Yes, as outlined in the guidance does not cover the broader topic of immunostimulatory oligonucleotides, as the considerations for those can be quite different.
Excellent. Thank you for that response. My next question is for Dr. Ramamurthy. If an ON is renally cleared more than 30% or greater than 30%, but is not intended in patients with moderate or severe renal impairment, is a full renal impairment study still required?
That's a good question. Though the oligonucleotide may not be intended for a renal indication, but if the final intended patient population who will ultimately be receiving the oligonucleotide therapeutic will include patients with varying renal functions, then we recommend having information on wide range of renal impairment. And we also recommend that conducting some of these analyses early in development because these can then inform on additional characterizations that may be needed, as well as the inclusion-exclusion criteria for late-phase trials. Thank you.
Excellent, thank you. I have another question for you, Dr. Ramamurthy. Why is FDA providing recommendations only on these 4 topics?
Thank you. That's another good question. FDA is issuing this guidance to provide consistent recommendations to sponsors focused on these 4 key areas that we just presented, for which we frequently receive questions from drug developers. And this includes characterizing QT, interval prolongation, performing immunogenicity risk assessment, characterizing the impact of hepatic and renal impairment, and the DDI liability assessment of that. And the guidance is largely based on recommendations that the agency had been already providing to individual sponsors. And we also recognize that as the field of oligonucleotide therapeutics progress, there are going to be innovations in, say, chemical modification of the bases, backbone, structural changes. There can be new delivery strategies, etc. So we encourage the sponsors to reach out to relevant divisions, review divisions, with specific questions concerning the topics that we just discussed. And we also encourage that some of these conversations can start as early as pre-IND applications as well. So we encourage you to take advantage of this mechanism to have a conversation. The recommendations provided are to ensure that there are consistent approaches that are utilized in the development of oligonucleotide therapeutic, taking into consideration their unique pharmacological and clinical pharmacology aspects. And the recommendations that are provided here were informed by the experience gained from FDA's review of these products during development, during regulatory approval, as well as in the post-market setting. It was also informed by a Federal Register notice that we had published a few years ago, asking questions that are relevant to clinical pharmacology consideration during the development of oligonucleotide therapeutic. And we published a draft guidance in 2022 and received comments from our stakeholders and other interested parties on this draft guidance. And these were all considered during the finalization of the guidance. And we are continuing to deepen and broaden our experience with this emergent therapeutic modality. And we will be planning to provide additional recommendations in the future on topics that may be relevant beyond the 4 that we just discussed. Thank you.
Of course, thank you. My last question for you, Dr. Ramamurthy. Do you know the general bioanalytical considerations guidance, or do you follow the general bioanalytical considerations guidance?
Yes, thank you, Corey, again for this question. We recommend that appropriate bioanalytical method should be used when characterizing the oligonucleotide therapeutic, as well as any of the relevant metabolites as applicable. And we recommend sponsors to use the FDA guidance titled M10, Bioanalytical Method Validation. That was published a couple of years ago. We request that you refer for additional details and general concepts to that guidance. Thank you.
Excellent, thank you. My next question is for Dr. Adeniyi. Are in vitro induction or inhibition studies needed?
Yes, thank you, Corey. So the in vitro inhibition studies are broken down. So in vitro studies for oligonucleotides as substrates are usually not needed. The guidance acknowledges that the disposition of oligonucleotide therapeutics are not anticipated to be affected by inhibitors or inducers of CYP enzymes, since the oligonucleotides are typically metabolized by endogenous nucleases. We're also not anticipating that modulation of efflux transporters like PGP, BCRP, the hepatic uptake transporters, or renal efflux and uptake transporters will have a significant impact on the pharmacokinetics of oligonucleotide therapeutics. And while the guidance does not actively dissuade performing studies, because this is an evolving field, companies can provide a justification to waive in vitro studies as a substrate. Now, in vitro studies to evaluate induction and inhibition studies are needed, but the guidance also affords companies the flexibility to provide justification if in vitro assessments are not to be conducted to evaluate the potential of the oligonucleotide therapeutics affecting CYP enzymes or transporters. If we gain enough experience or accumulate enough data to demonstrate that a certain type of oligonucleotide does not affect CYPs or transporters, and a certain investigational oligonucleotide therapeutic shares similarities, for example, in structure, as oligonucleotides where we've gained enough experience, then in vitro studies may not be needed for such an investigational oligonucleotide. Thank you.
Of course, thank you for that response. My next question for you is, what is the preferred in vitro system to evaluate inhibition potential of an oligonucleotide therapeutic?
Thanks again for this question. So the guidance notes that sponsors should carefully select an appropriate in vitro system to evaluate the drug interaction potentials, given that the differences amongst the in vitro systems have been reported. Now, there are publications that show some oligos inhibit CYP enzymes when using human liver microsomes, but the inhibitory potency significantly decreases when using hepatocytes to evaluate inhibition. So an explanation for this is that oligos may not be able to readily cross the cell membrane of hepatocytes, while experiments using microsomes, the oligos can directly assess the CYPs on the microsomes. So the presence of the cell membranes on hepatocytes may be better to recapitulate the permeability of oligonucleotides into hepatocytes in vivo. So companies can choose in vitro systems based on the scientific knowledge and their experience. We'll also encourage more research and publications to investigate the differences amongst systems and the underlying reasons, but we do not actively recommend one over the other. Thank you.
Excellent. Thank you, Dr. Adeniyi. My next questions are for Dr. Matabushi. What studies that are unique to each therapeutic modality development?
Thank you, Corey. And I think you're referring to my slide where I try to contrast between the different modalities. So in general, for small molecules where we have a rich history of understanding and development of various expectations for clinical pharmacology characterization, not all of those might apply for, say, oligonucleotides or monoclonal antibodies. For example, for small molecules, we routinely conduct a battery of renal hepatic impairment studies, drug-drug interaction studies, which are predominantly mediated through cytochrome P50 pathways. Some of those may not always apply to the same extent what we have in case of small molecules as it pertains to oligonucleotides, and definitely with monoclonal antibodies, we have a standalone guidance for drug interactions there, which takes into very unique considerations because those CYP pathways are not that directly relevant, and there are some indirect pathways. Things that are unique for oligonucleotides and monoclonal antibodies are the risk for immunogenicity, which is not something of a primary concern when we are talking about the small molecules, for example. And as it pertains to monoclonal antibodies, for example, based on the molecular weight, renal impairment studies might not be critical. There were some questions around what happens for conjugated oligonucleotides, and the same considerations could also apply based on the molecular weight. Now, with respect to oligonucleotides, some unique studies that might be of interest from a clinical pharmacology perspective would be pharmacodynamic characterization, especially given the disconnect between the time course of the pharmacokinetics and the pharmacodynamics. So in a nutshell, based on the distinctive features, the focus is going to be different across different modalities, while they may share some commonalities. Hope that helps answer the question.
Yes, thank you for that response. My next question for you, Dr. Matabushi. What is the role of model-informed drug development strategies in the development of oligonucleotide therapeutics?
Yeah, excellent question, Corey. The model-informed drug development approaches and strategies can be very informative in informing the drug development for oligonucleotides. And to follow up on the aspect that I touched upon on the pharmacodynamics, I'm going to reiterate that here and talk in terms of how model-informed drug development approaches could be useful. Given the temporal dissociation between the time course of the pharmacokinetics and the pharmacodynamic responses, as well as oligonucleotide treatment effects, I think it would be very useful to have a good characterization of the relationship and taking into account this temporal dissociation to be able to help inform dose selection, duration of studies, duration of the dosing interval, and things of that nature. You also heard from Dr. Ramamurthy's presentation that for oligonucleotides, one could utilize some of the quantitative approaches such as POP-PK, as well as exposure response relationships, if they are available across the entire spectrum, to be able to posit what might be the impacts of changes in the intrinsic factors such as renal impairment or hepatic impairment, and what, if any, those might translate into the responses, not just on the pharmacokinetics. So these, I think, are some of the areas where model-informed drug development approaches could be very informative for the development of oligonucleotide therapeutics.
Excellent. Thank you for that response, Dr. Matabushi. My next question is for Dr. Wu. Is there any evidence that an oligonucleotide blocks hERG? Are there any oligonucleotides that have demonstrated a positive QT clinical signal?
Thank you, Corey, for this question. So based on the oligonucleotides submitted to CDER, the answer is yes. We have seen direct and we have seen immediate actions of these molecules on the level of hERG channels to reduce or to enhance channel activity. Now, we don't have the clinical QT data yet, so we're still awaiting data to understand the non-clinical hERG clinical QT translation. Your second question is, are there oligonucleotides that have demonstrated a positive clinical QT signal? Well, a lot of our submissions are still in early stages. And so, again, we're still awaiting for clinical data, so not sure yet. But I can speak of those that have been approved. They're not associated with a positive clinical QT signal. Thank you.
Thank you, Dr. Wu, for that response. My next question for you, are thorough QT or TQT studies required?
Okay, so thank you again for this question. Oligonucleotide therapies are really emerging modality, and we're seeing an increasing number of submissions to the FDA. The current cardiac safety testing paradigm, for which the TQT study is a part of, was really developed based on adverse event mechanisms seen with small molecule drugs. And we know that oligos can differ in physical chemical properties from small molecules, which means that they can exhibit different off-target interactions, different off-target liabilities from small molecules. So I think it's really reasonable to ask this question about the utility and necessity of TQT studies for oligos. As Bart mentioned, oligos are very, very diverse, and these are comprised of chemically modified RNA or DNA products that differ greatly in structure, in size, in delivery strategy, conjugation to other moieties, and they can exhibit different chemical modifications to the base, to the sugar, and to the backbone. Also, their mechanisms of actions are very diverse, with some functioning just like proteins and others functioning via hybridization to affect gene expression. Now, what all these mean to us is that off-target interactions can differ greatly as well across different classes of oligos. That's why in this guidance, you see that an assessment of QT prolongation risk is still recommended. Now, having said that, there have been 17 of these products in different classes approved by the FDA, and our experience is, of course, accumulating how to approach this QT assessment for oligos, as well as other novel modalities, including amino acid products, given this accumulation of knowledge is actually a topic under discussion by the ICH E14 S7B expert working group. So I would say, please stay tuned to these two ICH level guidelines as well as this topic. Thank you.
Thank you very much for that response. My next question is for Dr. Rogers. Are the recommendations the same for ONTs administered intrathecally as those for those administered via IV?
Yes, thank you for the question. The recommendations are the same, and I want to point out that intrathecal oligonucleotides rapidly cross the blood-brain barrier to achieve systemic concentration. Oftentimes, concentrations in the kidney will exceed those in the CNS. So systemic exposure is expected, and the studies and requirements would be the same.
Excellent. Thank you for that response. My next question for you, Dr. Rogers. What studies might have to be conducted for individualized oligonucleotides for serious life-threatening diseases?
Yes, so these studies are separate, and they can be found in our what we call the suite of guidances for individualized antisense oligonucleotide products. They're beyond the scope today, but I do want to point you to the correct guidances that we have 3 draft guidances on this topic detailing the CMC, the non-clinical, as well as the clinical considerations for individualized antisense oligonucleotides.
Thank you for that response. And my third question for you, Dr. Rogers. In terms of immunogenicity, is it advised to further characterize anti-drug antibodies, for example, neutralizing antibody assays for ONT products?
So at this time, we typically do not ask for neutralizing anti-drug antibody assessment for these products, given their intracellular mechanism of action.
Excellent. Thank you for that response. And my last question for you, Dr. Rogers, if we don't see any abnormal PK or PD relation and no toxicity observed in preclinical, do we still need to do an ADA test for phase 2 samples?
Yes, in this particular instance, we would expect anti-drug antibody testing in the phase 2 studies. We also want to highlight the importance of anti-drug antibody testing in the later phase 2 and phase 3 pivotal studies, given the multiple dose nature of these studies, where we would expect anti-drug antibodies to manifest, opposed to the single dose studies often utilized in the first-in-human studies.
Excellent. Thank you for that response. My next question is for Dr. Ramamurthy. And this question is, should metabolites of oligonucleotides follow MIST guidance?
Thank you, Cody, for that question. We don't specifically touch on that in the guidance, but in general, in the oligonucleotide therapeutic program, they do major metabolites follow the MIST guidance recommendations. And I'll also point out to the draft guidance that was published fairly recently within the past month by FDA on non-clinical safety assessment. I would also encourage the viewers to go check out that guidance. Thank you.
Excellent. Thank you, Dr. Ramamurthy. My next question for you. Please share your view on radio-labeled oligonucleotide studies.
Thank you again for that question. I'm going to touch on 2 things in here because I'm not exactly sure what the question refers to. One is the mass balance studies. I will refer folks to the mass balance, human radiolabel mass balance studies guidance, final guidance that was published earlier in the year. We do say that when drugs such as oligonucleotide therapeutics that have known metabolism excretion pathway based on the pharmacology, basic pharmacology, non-clinical ADME information, then we do not expect to see changes in ADME properties. Then we do not recommend a mass balance study in such scenarios unless ADME properties are changing. And Dr. Rogers referred to this as well earlier. Another way to interpret this question is that, are there microtracer studies? And yes, we have seen microtracer studies for some oligonucleotide therapeutics that have been approved. Thank you.
Excellent. Thank you for that response. And my next question for you, Dr. Ramamurthy. Are the metabolites of oligonucleotide therapeutics active? Are there specific considerations related to the metabolites?
Thank you again for this question. This may be very specific to the oligonucleotide therapeutic in question. Unlike small molecules that are mainly metabolized by cytochrome P450s or other mechanisms, oligonucleotide therapeutics are metabolized by exo and endonucleases and by nuclease-mediated cleavage of the phosphodiester bond. And this cleavage happens predominantly in the 3' terminus and also at the 5' terminus by these nuclease-mediated cleavage events. The chain-shortened metabolites are these N-1, N-2 metabolites. And for some drugs, these metabolites can be pharmacologically active, sometimes even as much as the parent. And additional characterization may be needed for these metabolites. And again, I will refer to the recently published non-clinical safety assessment draft guidance as well for reference. Of the oligonucleotide therapeutics that have been approved, there are examples where chain-shortened metabolites have been considered as major metabolites and have been pharmacologically active, and these have been included in the subsequent ClinPharm analysis. Thank you.
Thank you for that response. My next question is for Dr. Adeniyi. Are in vitro studies for oligonucleotide therapeutics as substrate needed?
Thank you for that question, Kuri. I believe I addressed part of it in my earlier response, and the answer is they are usually not needed, but the guidance does not actively discourage performing the studies because it's an evolving field. So companies can provide justification if they don't want to perform studies to evaluate oligonucleotide as substrate. Thanks.
Thank you for that response. My next question for you, is there a role for PK modeling to assess DDI risk of oligonucleotide therapeutics, or are there recommendations on the use of DDI risk assessment models?
Thank you, Corey. There's limited experience with the application of PK/PD for DDI risk assessments of oligonucleotides, so it's not covered in the guidance. We also have very limited data to make any recommendations on risk assessment for oligonucleotide-specific models, especially considering that we have a paucity of data from in vivo DDI studies for oligonucleotides. Thank you.
Excellent, thank you. My next question is for Dr. Matabushi. Does the ICH M12 guidance apply for the development of oligonucleotide therapeutics?
Thank you, Corey. Excellent question. So the recently finalized ICH M12 guidance clearly states that oligonucleotides are beyond the scope of the guidance that was finalized. Primarily, that the ICH M12 was aimed at small molecules, and it states that regional guidances should be followed for specific clinical pharmacology considerations as well as the drug-drug interaction considerations on the topic.
Excellent. Thank you for that response, Dr. Matabushi. My next question for you. What is the minimum number of patients for hepatic or renal impairment in population PK analysis to be meaningful included in the label?
That's an excellent question, and we have heard a question of this nature previously also when it comes to utilizing population pharmacokinetic approaches. The way I would approach is that there is no a priori set minimum number of patients that is required, but what would be very important is to be meaningful, to be included in the label, the number of patients included and the range of the renal or hepatic function would become very important, and the number of subjects would be based on the ability to provide a precise and accurate characterization, which is very critical for considering what that effects are. I would actually have folks consult our final guidance on the population pharmacokinetics for more specific information on this topic.
Excellent. Thank you for that response. My next question is for Dr. Wu. What specific non-clinical assessments are needed for cardiac safety assessments?
Thank you very much for this question. So CDER recently released a draft guidance called the Non-Clinical Safety Assessment of Oligonucleotide-Based Therapeutics. And this guidance states that safety pharmacology evaluation of oligonucleotide products should address vital effects on vital functions, which includes cardiovascular system. And the draft guidance also refers to ICH S7A guideline. So small molecule drugs typically cause QT prolongation by blocking hERG channels in the heart. And the CDER draft guidance indicate that the hERG assay is not warranted for the oligos that belong to well-characterized class that has demonstrated negligible ability to block hERG channels. The class here means oligos that have conserved backbones, the sugar or other subunits, or targeting or other chemical modifications differing from one another only in the base sequence. That's a class. So the well-characterized class, you wouldn't need a hERG assay. The in vivo cardiovascular safety pharmacology assessment, which in vivo QT assessment, should still be conducted for all oligo products before clinical administration. Thank you.
Excellent. Thank you, Dr. Wu, for that response. My next question is for Dr. Rogers. Do we need ADME studies for oligonucleotide therapeutics?
As I touched on earlier, it depends on the diversity of the chemical class. So if it's a novel moiety where we don't have, it's not a well-characterized chemical class, and we don't have prior examples of, then typically, yes, we're going to need more in-depth ADME there. Whereas if it's a well-characterized class, chemical class, we do not typically need the ADME studies.
Excellent. Thank you for that response. My next question for you, Dr. Rogers. Are there any specific considerations or recommendations for pharmacodynamic or PD biomarker selection?
Yes, this is an excellent question and excellent topic. PD biomarkers are pivotal in our understanding of the mechanism of action, proof of concept, as well as guiding our need for dose escalation and dose finding. So we encourage sponsors to begin development of PD assays early on and to interact with the agency early on in their assays for these pharmacodynamic biomarkers, as again, their pivotal importance in the development of these products.
Excellent. And I think we have time for one more question today for you, Dr. Rogers. My final question is, are neutralizing antibody assays needed?
So typically, neutralizing antibody assays are not needed for this class of product. Again, this may change in the future, but given their intracellular mechanism of action, we typically do not ask for neutralizing drug antibody assays.
Excellent. Well, it looks like that is all the questions that we have time for today. Thank you again to our presenters and panelists for all of the excellent information. And now it is my pleasure to introduce our second presentation for today. And so first we will have our presenters, Dr. Sarah Ridge, a policy analyst on the guidance and policy team, within OCP, also an OTS at CDER at FDA. And then we will also hear from Chin Sun, Dr. Chin Sun, Biologics Lead in the Therapeutic Biologics Program, also within OCP in the Office of Translational Sciences here at CDER at FDA. Please join me in welcoming our first presenter, Dr. Sarah Ridge.
Thank you, Corey, for the introduction. My name is Sarah Ridge, and I will begin our discussion on the guidance entitled Clinical Pharmacology Considerations for Antibody-Drug Conjugates. I will be presenting the first part of this session. This will include an overview of the background of antibody-drug conjugates, or ADCs, and the guidance development. I will then discuss some of the contents of the guidance, including a description of the key considerations for ADC dosing strategies, the bioanalytical approach, and dose and exposure response. My colleague Chin Sun will take it from there and provide an outline of the key considerations for intrinsic factors, QTc assessment, immunogenicity, and drug interactions, or DDIs. She will then close out with some conclusions. After our presentation is ended, we will have a panel discussion where we will welcome our OCP colleague, Brian Booth, to join us in answering your questions. So ADCs have unique structure and are generally composed of a small molecule drug, also known as a payload, and an antibody, or antibody fragment, conjugated by a chemical linker. As you may be aware, ADCs with novel structures are in development, and the current guidance may not be applicable to these alternate structures. For example, novel payloads such as peptides, oligonucleotides, or toxins may have different absorption, distribution, metabolism, and excretion profiles, and may need different clinical pharmacology considerations. The figure to the right of the slide demonstrates the mechanism of action of the ADC. The ADC binds to the target antigen, gets internalized and degraded with the release of the cytotoxic payload, which leads to apoptosis of the target cell. To date, 11 ADCs have been approved by the FDA, both for solid tumors and hematologic cancer. This table breaks down each approved ADC by the antibody target, linker type, payload, payload target, as well as the drug-to-antibody ratio, or DAR. Guidance documents contain FDA's current best thinking on a particular topic. The Clinical Pharmacology Considerations for ADCs guidance was first published in 2022 as a draft, and was then published as a final guidance in March of this year. During this session, we'll be discussing the contents of the guidance. I'd also like to highlight the snapshot and podcast that was released shortly after the final guidance was published. We hope that these different forms of communication will facilitate awareness and engagement with this topic. The snapshot and guidance describing clinical pharmacology considerations for ADCs can be found on fda.gov. And so, before we get into the nitty-gritty of the guidance contents, I will first go over some of the key terminology. ADC is defined in the guidance as an antibody or antibody fragment conjugated to the payload via chemical linker. The total antibody refers to the collection of antibodies that are both unconjugated and conjugated. The unconjugated antibody is a free antibody not conjugated to any payload molecules. And the unconjugated payload is a free small molecule drug that is not conjugated to an antibody. The chemical linker refers to the linkage between the payload and the antibody. And the constituent parts of the ADC includes the total antibody, the unconjugated payload, the pharmacologically active metabolites, and the ADC catabolized products. And pharmacologically active metabolite refers to a pharmacologically active metabolite from the metabolism of the unconjugated payload that impacts the efficacy and safety profile. Now I will discuss the key considerations for ADC dosing strategies. ADCs combine the selectivity of an antibody for a specific target with the potency of a small molecule drug. Both the payload and antibody can independently contribute to safety and/or efficacy. It's worth noting that from a safety perspective, a relatively small increase in systemic exposure of the payload, as well as the ADC itself, can cause adverse reactions that are dose-limiting. And so when determining dosing strategies, a thorough understanding of the pharmacokinetics and pharmacodynamics of the ADC and its constituent parts early in development and the relationship to safety and efficacy outcomes is crucial to optimize the ADC dosage. Knowledge gained early in development facilitates the selection of an optimal dosage regimen later in development. A broad dosage range is recommended in early clinical studies that includes multiple dose levels and dosing regimens, such as single fractionated dosing. Consideration can also be made for both fixed and weight-based dosing if appropriate. It is important to note that exposure-response analysis in early development can be highly informative to dosing regimen selection in later development. Exposure-response analyses can also be used to select dosing strategies for patients in pivotal studies such as those with organ impairment. Now I will move on to describe recommendations for the bioanalytical approach. I would first like to note that all bioanalytical methods should be validated. More detailed information can be found on this in the FDA guidance that was published in 2022 entitled M10 Bioanalytical Method Validation. We recommend in the ADC guidance that the ADC and its constituent parts are measured in first-in-human studies with validated assays. In later development, the ADC and its constituent parts that are quantifiable in systemic circulation should be measured to inform or improve exposure-response analyses. Taking this information from early development into account, considerations can be made to exclude ADC constituent parts later in development. These considerations include the PK characteristics of each constituent part, for example, the correlation between the total antibody and the ADC concentrations, or whether systemic exposure of the constituent part is quantifiable. Next are the relevant non-clinical pharmacology, PK, or safety data. For example, this would include non-clinical data on the mechanism of action of the ADC, or the pharmacologic activity of the unconjugated payload, or metabolites. Another consideration is the preliminary exposure-response data on the contribution of the ADC's constituent parts to safety and/or efficacy. It is also worth noting that antibody target can shed into systemic circulation, and if this occurs to a significant extent, bioanalytical assays that distinguish the target unbound ADC from the target bound ADC should be employed. I will now discuss the key considerations for exposure-response analysis. As mentioned in a previous slide, exposure-response analyses can inform dosage selection and dosage adjustments. While we recommend conducting exposure-response analysis on the ADC and its relevant constituent parts early in development, justification can be provided for not conducting exposure-response analyses with an ADC constituent part later in development. Considerations to not include a constituent part would be the systemic exposure of the payload or pharmacologically active metabolites, the pharmacological activity of the antibody, and the correlation between the total antibody and the ADC concentrations. There are also factors to consider if the antibody target is known to shed into systemic circulation to a clinically meaningful extent. Exposure-response analyses can be conducted with the ADC and/or the total antibody that is not bound to the shed target in circulation. Considerations for such analyses can include the relative concentrations of the target-bound ADC compared to the target-unbound ADC in circulation, correlations between the target-bound ADC and target-unbound ADC concentrations, and the potential for the target-bound ADC to retain pharmacological activity. And this concludes my portion of this presentation. I will now pass it on to Chin.
Thanks, Sarah. Hello, everyone. In the following slides, I will discuss clinical pharmacology considerations for assessment of intrinsic factors QTc, immunogenicity, and DDI. First, let's see how to evaluate intrinsic factors in ADC development programs. Any intrinsic factor that has the potential to affect exposure of ADC or its constituent parts should be evaluated. For example, renal or hepatic impairment, pharmacogenomics, body weight, age, etc. These intrinsic factors can be evaluated through population PK analysis or in dedicated studies. The ADC guidance provides special considerations regarding organ impairment. First, organ impairment can impact the exposure of unconjugated payload, and the principles are similar as for small molecule drugs. FDA has relevant guidance documents, and the revised renal impairment guidance was published in March 2024. The hepatic impairment guidance was published in 2003. There are some new considerations regarding the impact of organ impairment on ADC's antibody part. Assessing the impact of organ impairment on exposure of ADC or total antibody could be relevant under certain scenarios. For example, for renal impairment, ADC could be eliminated through renal route if an antibody fragment is used and the molecular weight is less than 69 kilodalton. For hepatic impairment, decreased ADC or total antibody exposure has been reported in literature, which could potentially impact efficacy. Similar for small molecule drugs, hepatic impairment can increase exposure of unconjugated payload, which could impact safety. Now let's see study approach for organ impairment. First, we will discuss when to choose population PK analysis. One scenario will be if patients with organ impairment have been enrolled in pivotal studies and the PK, safety, efficacy information are available. To inform inclusion of specific populations, sufficient ADME information from non-clinical and early clinical studies are needed to ensure patient safety. In addition, there should be adequate PK sampling and a sufficient subject number to support population PK analysis. Besides PK data, there should be relevant safety and efficacy information in patients with organ impairment to understand the impact of an exposure change, if any. The number of enrolled patients with organ impairment to provide sufficient safety and efficacy information should be prospectively discussed with the FDA on a case-by-case basis. Next, let's see when to choose a dedicated study. The following information should be considered: ADME and potential changes in systemic exposure of ADC and the unconjugated payload. For example, expectation of a clinically significant change in the systemic exposure of the unconjugated payload could inform the lead for a dedicated study. Dose exposure-response relationship for efficacy and safety, and the anticipated clinical consequence of the systemic exposure change. Also, safety signals that can be correlated with exposure changes especially those detected in patients with organ impairment. After organ impairment, now let's see the considerations for another intrinsic factor, pharmacogenomics. For ADCs, considerations for pharmacogenetic evaluation depends on ADME information, systemic exposure of the unconjugated payload, and the role of antibody in mechanism of action. For example, genetic variants and/or expression of antibody target can impact patient's response to ADC. Genetic variants of metabolic enzymes and transporters can impact clearance of unconjugated payload, such as CYP2D6, UGT1A1, BCRP. Genetic variants of Fc gamma receptors can affect ADC binding to Fc gamma receptors, leading to altered ADCC or ADCP activity when they contribute to mechanism of action. We have talked a lot about intrinsic factors. Now let's switch gears to discuss considerations for QTC assessment. QT assessment is recommended for all ADC development programs, and it should focus on the small molecule part. The antibody part generally has a low risk unless it is suggested by mechanistic considerations or by data available from clinical or non-clinical studies. For the unconjugated payload, the principles for characterizing QT prolongation risk are similar to those for small molecule drugs. The relevant FDA guidance documents are included here, including E14 Clinical Evaluation of QT, E14 Questions and Answers, and also E14 and S7B Clinical and Non-Clinical Evaluation of QT, Questions and Answers. Next, let's see immunogenicity assessment for ADCs. The immune response can be generated to any part of the ADC, such as antibody, payload, or the linker. ADC development programs need to evaluate immunogenicity and its impact on PK efficacy and safety. A multi-tiered immunogenicity assessment is recommended based on FDA guidance, including screening, confirmation, neutralizing antibody, and a titer test. In addition, multiple assays may be needed to evaluate where ADA binds to. The relevant guidance documents are listed here, including immunogenicity assessment for therapeutic protein products published in 2014, immunogenicity testing of therapeutic protein products, developing and validating assays for anti-drug antibody detection, published in 2019. DDI is another challenging topic needs to be addressed in ADC's development program. This slide summarizes some general considerations for payload DDI evaluation. I will not go through one by one, but just highlight a few for your attention. First, characterizing systemic exposure of unconjugated payload, although possibly low, is important for determining its potential as a precipitant. Second, in vivo DDI evaluation of cytotoxic payload as a substrate from inhibitor is recommended if any DDI risk is identified from in vitro assays. However, in vivo DDI evaluation with inducers may only be relevant if a payload has significant exposure and contributes to efficacy as the ADC. Under certain scenarios, DDI may need to be evaluated for the antibody part. For example, when ADC is administered with medications that share the same PD target, medications that interfere with FcRn binding, immunosuppressors if ADC's PK are affected by immunogenicity. Although there could be limitations in ADC dose modification, it is important to understand whether other drugs can be safely used with ADC and to inform the labeling strategy. Here are the conclusions for today's presentation. ADCs are complex products, and their constituent parts can independently contribute to efficacy and safety. In early development, a broad dosage range evaluation is valuable to inform dose selection for later studies. The assay validation and analyzing certain ADC constituent parts early in drug development would lead to an efficient program. Those exposure response analyses are important to guide dose strategy at different clinical stages. Evaluation of intrinsic and extrinsic factors is critical for therapeutic individualization and labeling recommendations. QTC and immunogenicity assessments are recommended for all ADC programs to assess the impact and to inform risk mitigation strategy. Also, knowledge sharing and collaboration among key stakeholders are important to drive efficient ADC programs. This slide provides some helpful resources for FDA guidance snapshot, and a podcast. Also, drugs at FDA with product labeling and FDA review summary information. Finally, acknowledgements. First, we want to thank FDA OCP ADC Guidance Working Group listed here for their tremendous efforts and contribution. We also want to thank all our current and former OCP colleagues who contributed to this ADC guidance development. The last but not the least, thank all the key stakeholders who provided valuable comments to help the guidance development. Now, let's do a quick knowledge check. The question is, what are the key clinical pharmacology considerations for ADC programs? A. Exposure response analysis and dose selection. B. Intrinsic, extrinsic factors. C. Immunogenicity, QTC assessment. D. All of the above. I will pause a few seconds for you to think. The answer is D. Now let's welcome Brian to join us, and we will be happy to address any questions you have. Thank you so much.
Thank you again to our presenters today for their excellent presentations. We will now begin our second question and answer panel. If you haven't had a chance to enter your question into the Q&A chat box, please do so now. We will answer as many questions as time allows. Today, we will have our presenters who just spoke, Dr. Sarah Ridge and Dr. Chin Sun. They will also be joined by Dr. Brian Booth, Director in the Division of Cancer Pharmacology, also within OCP and OTS at CDER at FDA. Now, our first question today will be directed towards Dr. Ridge as well as Dr. Booth. What clinical pharmacology principles of ADC characterization, if any, are also applicable to the relatively new therapeutic modality of antibody radiopharmaceutical conjugates, or ARCs? Is the agency developing its thinking on the development considerations for ARCs?
Good afternoon, everyone. This is Brian. Thank you for the question, Corey. Very interesting one, actually, because certainly the ARCs are structurally a lot like ADCs, right? They have an antibody modality to target specific antigens, so they can hit tumor cells very specifically, and they have a payload. But there are some important differences there too, right? Because here the payload is a radioactive nuclide. And usually it's just a nuclide and not a big molecule like MMAE that's attached by a linker. So there's some differences there as well. And so would the ADC guidance apply to that? Well, it can for some of the things, but for some other parts it probably wouldn't. And if you look across our guidances, you'll see that we really use the same sort of conserved principles or issues across all, look at exposure response, you want to look at immunogenicity for antibodies, renal, hepatic, drug-drug interactions for small molecules and things like that. So there would be some of these things in the ADC guidance that would apply here, right? So some of the exposure response and early dose finding sorts of things, but maybe not so much things like renal or hepatic impairment or maybe the drug-drug interactions if we're looking at a radionuclide, right? Because those wouldn't really apply necessarily. On the other hand, you'd have to do or look at some different things, right? So you're looking at some radioactive nuclides, so you have to consider things early on, like looking at the dosimetry to find out which organ systems are going to be sensitive to this particular radionuclide, and that'll give you some insight in terms of the dosing range you're going to start looking at. And you'll have to consider the overall radiation exposure in the dose selection that goes along with this. So is the agency developing its thinking on the development? Yeah, I think it's safe to say that. And I think probably likely you'll see some further communication on that future. Thank you.
Excellent. Thank you for that response. I have another question for you, Dr. Booth. Should the concentrations reported by the ADC PK assay and the total antibody assay be similar? If so, how similar is considered similar enough?
Okay, thank you for the question. So, let me answer the second question first. So how similar is similar enough? Well, we don't really have a criterion that we attach to that, right? What we see in practice generally is that the total antibody parallels the ADC concentrations over time, and they're generally very close to each other. You would generally see some bias, some consistent bias between them over the whole time period of measurement. And sometimes what happens is at the end, you might start to see a bit of a difference between the two curves. But we don't have a specific criterion for that. So, and the how similar they are is just sort of described qualitatively rather than quantitatively. Thank you.
Excellent. Thank you for that response. My next question is for Dr. Ridge. For aptamer-drug conjugates, drugs, should we follow the guidance of ADC or oligonucleotide in preclinical development?
Hi, Corey. Thank you for this question. So the guidance doesn't specifically cover aptamer-drug conjugates. However, in the absence of a specific document, many of the principles in the ADC guidance would generally apply. So it'd be reasonable to follow insofar as it applies. I'd also like to note that as far as the oligonucleotide guidance, aptamers are out of scope in that one. Thank you for the question.
Of course, thank you. My next question for you, Dr. Ridge. What analytes should be measured in ADC development program? Are there different considerations for early stage versus late stage?
Sure, thank you for this question. So at a minimum, the ADC, the unconjugated payload, and the total antibody should be measured in early stage clinical development. Pharmacologically active metabolites and catabolites should also be included if they're relevant. A thorough understanding of the PK and PD of these analytes early in development, they do inform decision-making later in development, such as selecting the optimal dose. And so using the data from early stage clinical studies, justification can be made to waive the measurement of an ADC constituent part later in development. So for example, measurement of the total antibody can be waived if the ADC and total antibody are shown to be highly correlated, or if an analyte such as the unconjugated payload cannot be measured using a sufficiently sensitive assay. Thank you.
Excellent. Thank you for that response. And one more question for you, Dr. Ridge. Is it necessary to conduct ADME or PK studies for the payload if the payload was already used in the approved ADC?
So the sponsor would need to own or have a right of reference of the approved ADC product in order to use the information from that development program in a standalone study. So if the sponsor does not have right of reference, an assessment of or prediction of human elimination pathways of the payload should be made. Thank you.
Thank you. My next question is for Dr. Sun. When during clinical development, should pharmacogenomics be assessed? Already during phase 1A and expanded cohorts 1B, or during phase 2 or 3?
Yeah, hi, thank you so much for this great question. And if the sponsor during the ADME characterization already found some, the cytotoxic payload is a subject for some polymorphism enzymes, and we will recommend them to evaluate the pharmacogenomics information early in clinical study because the information collected will be important to inform the study design, such as inclusion, exclusion for later studies. That also important for them to include some risk mitigation strategy to ensure the patient safety, especially consider the cytotoxic of the payload. Thank you.
Excellent. Thank you for that response. My next question for you, Dr. Sun. In case of a low immunogenicity risk ADC, is it okay to have immunogenicity as an exploratory objective instead of a secondary in a first-in-human trial?
Yeah, Corey, thank you again for this good question. And the sponsor mentioned, this question mentioned for the first-in-human, assume it's a low immunogenicity risk ADC. And we think the sponsor need to provide a thorough justification. If no clinical data, why do they think this ADC is a low immunogenicity risk? If it is justified, and I think included as exploratory or secondary can be justified, they can evaluate thoroughly in later study to confirm the immunogenicity is low. Thank you.
Thank you, Dr. Sun. My next question for you is, if systemic exposure of unconjugated payload is low, is there a need to evaluate its DDI risk as an inhibitor for transporters?
Yeah, thank you for this good question. For in vitro DDI characterization, the quick answer is yes. And the need of in vivo DDI study will depend on the predicted risk. The current ADC guidance recommends ADC development program should include in vitro DDI assessment using both CYP enzyme and transporter-related assays. In addition, the systemic exposure of unconjugated payload, although possibly low, is important for determining its DDI potential as inhibitor or inducer. So the need of in vivo DDI study can be evaluated based on FDA DDI guidance published in 2020, including in vitro drug interaction studies and clinical drug interaction studies. Thank you.
Thank you for that response. My next question is for Dr. Booth. We have seen some DDI study waivers for recent MMAE-based ADCs based on past available clinical DDI data and PBPK. Can the sponsor leverage learnings from an ADC with the same linker and payload? Do you have any thoughts on the acceptance of PBPK for assessing DDI for novel payloads?
Thank you for the question. So yeah, we have indeed seen some cases where we've issued some waivers because we already had information, related to MMAE, and so they were using the same payload and the same linker, and it was reasonable to leverage that information. But I think Dr. Ridge mentioned rightly that one of the important considerations there is you're going to have the right of reference to that information in order to use it. And if that's the case, then it's certainly worth having a conversation with the review team in similar kinds of situations. And the second part of the question was, oh, can we use PBPK? Well, we use PBPK for DDI quite a lot, right? Particularly with small molecule drugs. And so it's a very well-used modality for that. And so there is some promise that you can use it here, but I think the big key here is going to be whether you have enough information to characterize the behavior of the novel payload. And if you don't have that, you won't be able to develop the models that are going to work very well for this purpose. So it depends a lot on the amount and the kind of information you have around that. Thank you.
Of course, thank you, Dr. Booth. My next question for you: how stringent is FDA with dose optimization, dose and schedule, for ADCs in early clinical development versus post-approval?
That's another good question. So, we launched this Project Optimus initiative, I guess, a couple of years ago now. And the focus of that is to push dose selection, dose exploration earlier in development to try to get a better handle on the dose that offers us the best efficacy with the least amount of toxicity. And we did that largely because in our history, we have pursued this MTD approach, which sacrifices that therapeutic window for the sake of maximizing efficacy, which get a lot of toxicity. And what we found over the years is we have a lot of drugs that are labeled at doses that they're just very toxic, right? And we see in the pivotal trials and in the post-approval setting, there's a lot of adverse events, and these lead to dose discontinuations, dose reductions, and dose delays, and those kinds of things. And so we have a lot of drugs that are problematic in that way. And in our efforts to do more dose exploration or dose selection in the post-approval setting, that's been fraught with all kinds of difficulties, not the least of which is that the therapeutic landscape changes so quickly, right? And then it becomes very hard to enroll patients into these studies. And so you're left with studies that are incomplete, and you don't get the answers about what is the better dose for that particular product. So it's been very difficult to do that. And that's why we are now focusing on trying to get that answer early in development. And it's not just early in development, it's actually through development, right? But we start that effort early in development to try to get the answer there. And when we're talking about oncology, we're thinking this way for all therapeutic modalities, like small molecule drugs, antibodies, bispecifics, and ADCs. We view that all the same way. So we would really encourage people to put the effort early on into trying to find the dose that's best for the patients. Thank you.
Thank you for that response. My next question is for Dr. Ridge. Is there a plan to provide specific guidance for peptide drug conjugates? If not, is there applicability of the ADC guideline?
Thank you for the question. So I can't speak to future plans for guidance development. However, while peptide drug conjugates are out of scope for this guidance, many of the principles would still apply, and so careful consideration can be made there. And I'd also like to direct you to the draft guidance clinical pharmacology considerations for peptide drug products for peptide specific considerations. Thank you, Corey.
Thank you for that response. My next question for you, Dr. Ridge. The newer generation of ADCs are pretty stable in circulation. It may be very challenging to detect or measure the released payload or metabolites. Would FDA still require measurement of pharmacologically active components?
So we would recommend using a sufficiently sensitive assay to make efforts to measure the payload and pharmacologically active components early in development. Having said that, if they're not measurable, well, then they're not measurable. And so we do not recommend continuation for, let's say, exposure response or dose selection using an unmeasurable payload. Thank you.
Excellent. Thank you for that response. And my next question for you, Dr. Ridge, is there a need to conduct mass balance study for ADCs?
Good question. So monoclonal antibodies are typically eliminated via intracellular lysosomal degradation to amino acids or other mechanisms. And so human mass balance studies may not be feasible for the ADC. However, an assessment or prediction of human elimination pathways of the payload can be made that would include an assessment of excreted metabolites in urine or feces. This can be conducted in early clinical trials, animal studies, and/or in vitro assays of the payload. And just to note here that the ADME profile of the payload should be used to enroll patients based on various intrinsic and extrinsic factors with the safety and efficacy profile of the ADC. Thank you.
Thank you for that response. My next question is for Dr. Sun. If unconjugated payload is a CYP3A4 substrate, is there a need to evaluate in vivo DDI with CYP3A4 inducers?
Yeah, thank you for this great question. As mentioned in the presentation, in vivo DDI evaluation of a payload as a substrate from inducers may only be relevant if the payload has significant systemic exposure and contributes to efficacy as an ADC. For most approved ADCs, the systemic exposure of unconjugated payload is very low, and a further decrease will not impact the efficacy. Usually, the exposure of unconjugated payload in tumor cells contributes to the efficacy, while its systemic exposure correlates with safety. Thank you.
Excellent. Thank you for that response. My next question for you, Dr. Sun. For ADCs with a low ADA incidence and no impact on PK, efficacy, or safety, is there a need to evaluate epitope specificity for immunogenicity assessment?
Yeah, thank you for this question. The quick answer is no. Based on immunogenicity testing guidance published in 2019, a multi-tiered immunogenicity assessment is recommended, including screening, confirmation, neutralizing antibody, and a titer test. However, further evaluation, including epitope or domain specificity test, is risk-based. For the low-risk product mentioned here, it may not be needed for further assays. The sponsors are encouraged to discuss their proposal and rationales with the agency. Thank you.
Thank you for that response, Dr. Sun. My next question is for Dr. Booth. Can you use literature information to support ADC development and BLA filings?
Hello again. Thank you for the question. I think in theory it's possible we could use some literature. Actually, I have to check myself. There are some specific requirements around what's permitted for BLAs, and ADCs would fall under that. And there are specific requirements that the information that gets used in the labeling for the products is actually owned by the group that's developing the particular product. So it's sometimes very difficult to borrow information about things across different products with BLAs, and we can run into some difficulty there. I know that we sometimes use literature information for small molecules where the regulations are a little bit different. So I think there are probably some limited cases where we can do that, but I think there are times where that's problematic for a number of reasons. Thank you.
Thank you for that response. My next question for you, Dr. Booth, is there a need to conduct biodistribution studies of ADCs using radio-labeled PET active molecules?
All right. So I presume this is an ADC that's labeled with the PET nuclide. And as I alluded to earlier, these kinds of molecules, the ARCs, are going to sort of borrow bits and pieces from some of the other things we've already talked about, right? So like some of the antibody type of behavior has to be characterized appropriately. And when we're using these radio labels, we have to characterize those appropriately as well. I spoke earlier about conducting dosimetry studies with some of these products to identify which of the organ systems are going to be at greatest risk from these products. And in the same vein, we often do biodistribution studies to assess what's happening with the radionuclides. Thank you.
Thank you for that response. My next question is for Dr. Ridge. Dosing for ADCs has thus far been body weight or surface area based. In your opinion, why have sponsors of ADCs in the oncology space not opted for flat dosing yet?
Thank you, Corey. This is a good question. So I think it may be related to the cytotoxic effect of the payload and the narrow therapeutic index of ADCs. Body weight normalized dosing may help reduce exposure variability and lead to better safety profile of ADCs. And so in a recent ACCP annual meeting, and also as indicated by some published literature, it suggested to reconsider the value of body weight dosing when the power exponent of the body weight effect on clearance or central volume of distribution is under 0.5. In that scenario, fixed dosing may result in less PK variability than body weight normalized dose. However, it was pointed out during these discussions that the analysis was based on limited products, and investigators may still prefer to use body weight normalized dose. And so regarding this, and as well as dose capping, sponsors would need to have a good understanding of the variability and associated covariates, as well as the ER relationship for efficacy and safety to ensure patient safety. Thank you for the question.
Of course, thank you for that response. My next question for you, Dr. Ridge. In clinical trials, based on BLA reviews, sponsors mostly measure unconjugated payload, conjugated ADC, and total MAB, but not the linker alone or the payload attached to the linker. Do you see the necessity?
Yeah, so this is an interesting one. So far, I haven't seen cases where sponsors measure the linker alone. But we have seen a situation where the sponsor did measure the unconjugated payload together with the payload attached to the linker, because the AUC for the payload attached to the linker was comparable or even slightly higher compared to the unconjugated payload. So both in clinical trials and also they measure this in clinical trials and also the DDI risk in vitro for both. And so the important piece to keep in mind here is that gaining understanding of what the active and measurable components are early in development provides a clearer picture of what analytes are relevant for later in development. Thank you.
Thank you for that response. My next question is for Dr. Sun. Why do we need to worry about QT assessment for ADCs since payload exposure is usually very low?
Yeah, thank you, Corey, for this good question. For the unconjugated payload, the expectation and the principles for QT assessment are similar to those for small molecule drugs. Even if the payload exposure is low, it still needs to be evaluated clinically to confirm it has low risk for QT prolongation. For currently approved ADCs, although most of them had no prolongation risk based on their QT assessment, some ADC did show risk for increased QT and needs to include a risk mitigation strategy in their labeling. Therefore, QT assessment should be conducted for ADC development programs. Thank you.
Thank you for that response. My next question for you, Dr. Sun. Can PBPK be used for prediction of the impact of organ impairment, e.g., moderate or severe renal and hepatic impairment?
Yeah, thank you for this great question. Although PBPK modeling has been used to predict the impact of organ impairment, including moderate or severe renal hepatic impairment, those applications are mainly used to support the study design to evaluate their impact clinically. It is still challenging to use PBPK modeling to waive organ impairment assessment currently. Even for small molecule drugs, PBPK modeling has been reported to overpredict, sometimes also underpredict the exposure, especially when organ impairment severity increases. For ADCs, it is more challenging since organ impairment may affect both the antibody part and the small molecule part through different mechanisms. For example, hepatic impairment has been reported to decrease ADC or total antibody exposure, while increase the payload exposure. Therefore, there are lots of knowledge gap we still need to address to improve our understanding about the complex physiology and also biological changes for renal impairment or hepatic impairment before we can rely on PBPK modeling prediction. Thank you.
Thank you for that response. My next question is for Dr. Booth. Can goals be managed to accommodate FDA guidance on diverse recruitment?
All right. I think they're asking whether our diversity expectations would apply for ADCs. And so generally, I would say the answer to that is yes. And it doesn't really matter sort of what the therapeutic modality is. We strongly encourage sponsors who are developing products for oncology, as well as other therapeutic areas, really to ensure that they recruit an adequate number of patients in different demographic and ethnic groups, so that we have enough data to appropriately assess the behavior of the drugs in these different groups, so that we can give good, safe dosing instructions for those patients. Thank you.
Thank you for that response. My next question for you, Dr. Booth. How do you think MIDD approach can facilitate ADC development program?
That's a great question. Right, so we see the application of MIDD across a broad range of products, and in my opinion, it's been extremely helpful in helping to pick doses and dosing regimens for many products. We use it extensively in bispecific type of modalities, particularly for looking at things like step-up dosing to avoid CRS. And these leverage non-clinical data and then incorporate the clinical data increasingly as it becomes available. And they've developed these models that we've been able to use to simulate the behavior of these drugs under different conditions, and that's allowed us to help select doses for further testing in the patient populations to demonstrate safety and efficacy. So I think the MIDD approaches are fantastic, and I would encourage everybody to do it. Certainly, I know Dr. Matabushi has his open sign above his office for all sorts of applications along these lines. Thank you very much.
Thank you for that response. And my last question for you, Dr. Booth. For approved ADCs, lots of them do not have data for moderate or severe organ impairment. How do you address this knowledge or data gap?
Yeah, very good question. So, going back a little bit, I think we've talked about this a little bit. Often we see a sort of a two-part approach to this. Okay, so early or during development, well, we step back a little bit further. A few years ago, we issued a guidance that broadened inclusion criteria or eligibility criteria for trials, and that allows people to enroll patients with more or higher levels of organ dysfunction in their studies. And so what we've seen over the past few years with ADCs as well as small molecules and antibody drugs is the inclusion of patients who have mild and moderate impairment to some degree. So that gives us a glimpse of the behavior of the drugs in these kinds of patients. And with ADCs, we see that sometimes where they've included some of these patients in the efficacy trials. And in some cases, they have also supplemented this with some dedicated organ dysfunction studies at the renal or hepatic impairment. help us get the answer about how these things are behaving in these patients, right? And the concern, can be twofold. I think most often what we see is you see the ADC, the payload go up, and that's a concern because that's going to cause a lot of toxicity. And when we understand that behavior, what we do is we put in the labels that we should not administer these to patients with this level of organ dysfunction because of these toxicities. That's because it's too hard to manage dose adjustments in these kind of settings. Now, in the cases we see here, where there are some products that don't have information, I can tell you that there's probably a fair number of them that have outstanding post-marketing requirements, and they're doing studies to look at those things as we're moving forward. And I think there may be some cases here and there where there are no such things, where we don't have the information. And in those cases, we've just taken a very conservative approach in saying, do not administer these products to patients with these kinds of levels of organ dysfunction. Thank you.
Excellent. Thank you for that response, Dr. Booth. I think we have time for one more question today. This question is for Dr. Ridge. While I agree that ER analysis for safety should be conducted with all relevant analytes, efficacy is mostly relevant to ADC exposures, and analyses with other constituent parts become overburdensome. What are your thoughts on whether ER analysis for efficacy can be conducted with ADC only?
Thank you for this question. This is not necessarily the case for all products. For example, the unconjugated antibody can be pharmacologically active in some cases, and so exposure response analysis with the total antibody is relevant, or can be relevant in determining whether the antibody contributes to efficacy. So using knowledge gained from early exposure response analysis can be used to justify not conducting exposure response analysis with any ADC constituent part later in development. Thank you for this question.
Excellent. Thank you for that response. And that looks like that is all the questions that we have time for. Thank you again to our presenters for the excellent information. Now I have a few final reminders as we close out our day. First, the recordings will be posted on our website, www.fda.gov/CEDAR-SBIA. And again, please note that SBIA will be sending out a thank you email within 24 hours of today's presentation. Thank you again to all who participated today. This concludes our webinar session, and we hope that you will have a good rest of your day.